Hello. So I'm presenting these slides today on behalf of my colleague, Dr. Katherine, who unfortunately wasn't able to join us today. So deeply phenotyped biobanks such as UK Biobank allow us to study the genetic basis of thousands of different phenotypes within a single cohort. This hypothesis-free approach can benefit many different aspects of the drug development process. At the Centre for Genomics Research at Astrazeneca, we analyse genetic variation at two different levels. For variants that we see in more than a handful of participants, we can estimate their effects individually. But for variants that are more rare than that, in order to maximise power, we aggregate the effects of similar variants across the gene and perform analysis at the gene level. We apply these two approaches to binary and quantitative phenotypes, with the result that we end up with 4 sets of results being generated for every dataset analysis. We have previously applied this approach to a phenome-wide association study of coding variation in 450,000 exomes from UK Biobank, and at the gene level we were able to identify thousands of robust associations, many with large effects. We have made these results freely available to browse and download at this website, azphewas.com, in the hopes of empowering and giving back to the research community. This screenshot shows the gene-level results for rare variation and how they are associated with germline cancer predisposition. And you can see that this analysis is able to highlight 4 well-known breast cancer predisposition genes. So we know that the genome comprises just 1% or so of the exome, with other elements such as untranslated regions and introns having much larger footprints. These other elements may harbour novel drug targets, and these could potentially be targeted using modern drug modalities such as antisense oligonucleotides, small interfering RNAs, and therapeutic genome editing, among others. There are 3 important ways in which we think that genome sequencing improves upon exome sequencing. One is that it gives us better ability to detect variants in regions that are poorly covered by exome sequencing, such as GC-rich regions. In other words, it gives us a better exome. Secondly, it enables us to capture variants in non-coding regions, such as promoters, enhancers, and introns. And finally, it enables us to detect structural variants, many classes of which cannot be detected at all, and others of which can be detected only imperfectly using exome sequencing. So we've already heard today about how a consortium was formed in order to facilitate whole genome sequencing of 500,000 UK Biobank participants. And you've heard how this is one of the most ambitious sequencing efforts in history. So at the completion of this effort, we have 10 petabytes of compressed data. What needs to happen next in order to put this data in researchers' hands? Well, we need to align it, and we need to call variants. And we want to call not just small variants, but to take full advantage of having whole genome sequences to look at classes like structural and copy number variations. Of course, we need to be able to do this at a huge scale, efficiently and affordably, in a scalable way. So you can imagine this is quite some challenge, but it was embraced by my colleagues under the leadership of Sebastian Wasilewski, who have performed alignment and variant calling of these 500,000 genomes using the DRAGON platform. This produces not just small variant calls, but copy number variant calls, structural variants, some short tandem repeats, CYP2D6, and many other forms of output. And all of these outputs will be made available to the UK Biobank research community, we hope by the end of 2023. So the actual processing was performed on Amazon Web Services cloud computing, and at its peak, this effort was able to process, to align and variant call 20% of the entire data set in just 10 days. I'm now going to tell you a little bit about some of the work we have done looking at the first 200,000 genomes, some of which is mature and some of which is only just beginning. So I'm going to start with some work from my colleague Jonathan Mitchell, who has compared small variant calls between matched genomes and exomes of UK Biobank participants. And similar to our colleagues at Decode Genetics, He has demonstrated that WGS captures more coding variants than whole exome sequencing. This table shows that WGS is able to recall more variants detected in exome sequencing than exome sequencing can recall in the whole genome sequences. Jonathan then looked at Mendelian inconsistency rates in 92 trios in both SNPs and indels in the exomes and matched genomes. And you can see that the whole genome sequencing achieves lower Mendelian inconsistency rates than whole exome sequencing, while SNPs achieve lower Mendelian inconsistency rates than indels, suggesting that WGS is likely to produce fewer false positive calls than exome sequencing. I now want to tell you a bit about a phenome-wide association study of the untranslated regions of the genome, which has been led by my colleagues Kaushik, Kwon-Lee, Feng-Wan and Ryan. So we have looked at around 20,000 binary phenotypes and over 1,500 quantitative phenotypes. And we have used two different filters to define rare and potentially interesting variants in the untranslated regions, using firstly two different allele frequency cutoffs and then combining these with either the absence or presence of a filter for a score known as CAD, Combined Annotation Deficient Depletion Score. And we have considered variants across the 3' and 5' UTR combined for the moment, but we might separate these out later. So these Manhattan plots show the result for binary phenotypes at the top and quantitative phenotypes at the bottom. And we've had around 24 binary traits and over 200 quantitative traits reach significance in this association analysis. And I've highlighted here two positive control results, which is the beta hemoglobin locus and beta thalassemia, and apolipoprotein A5 and disorders of lipoprotein metabolism. And in fact, rare mutations in the untranslated regions of both of these genes have been linked to the corresponding phenotypes, validating this approach. Finally, I want to tell you a little bit about some work on the DRAGON whole genome copy number variation calls that's being undertaken by my colleague Zoe Zhu. So copy number variations are a form of structural variations. Within the autosomal regions of our genome, we usually have two copies of each gene. So this cartoon shows firstly what copy number loss might look like, either homozygous, copy number 0, or heterozygous, copy number 1, and then what copy number gain might look like if you gain 1 or 2 extra copies of a gene. Copy number variation is a really important class of variation, and so I wanted to show you a brief case study here, some work that was published by my colleague Katherine a number of years ago, showing how a pathogenic copy number variant that is easily visible using whole exome sequencing in the top panel is undetectable using whole exome sequencing at the bottom. So in combination with a point mutation in the same gene, this copy number variation is thought to be causing inherited retinal disease in this patient. Returning back to UK Biobank and the DRAGON calls, the top of this diagram shows you some of the steps that are undertaken by the DRAGON copy number variation caller. These result in VCF files of single sample copy number variant calls in yellow, and Zoe has then undertaken filtering and merging of these calls to attempt to produce a higher quality set of CNVs for analysis. So if you look at the plot at the bottom here, we can see that the typical participant has around 50 high-quality deletions, copy number losses detected, with fewer copy number gains detected. And the plots at the top show the size distributions for copy number gains and losses. So returning to the same set of 92 trios I told you about earlier, Zoe now looked at Mendelian inconsistency rates for copy number variations. So in red, you see what these look like for the pass calls output by the DRAGON copy number variation call. And in blue, you can see what they look like after the filtering and merging process. So this process reduces the Mendelian inconsistency rate down to around 3%. So the next step is to conduct a phenome-wide association study using this analysis, and I believe this has begun just in the last few days. For the moment, I want to show you that we are able to replicate one result which has been detected using array-based copy number variation calls from this same UK Biobank dataset. And this is an association between deletions on chromosome 16 and increased body mass index. So you can see the carriers of the 50-odd carriers of this deletion in red compared to non-carriers of this deletion in blue, and you can see that these have a higher body mass index distribution overall. So like all the rest of the DRAGON CNV calls, these will be made available to the research community, and we think this is likely to be at the end of the year. So the scale and biobank of UK Biobank makes it an invaluable resource for drug development, and the UK Biobank Whole Genome Sequencing Consortium has undertaken sequencing and data processing at an unprecedented scale in order to generate data that will be available to the research community. Whole genome sequencing allows us to expand our PheWAS and other types of research to include structural variations, non-coding sequence, and also coding variants that might be missed by exome sequencing. Future work within the Centre for Genomics Research this year will involve continuing our analyses and expanding these to the full 500,000 genomes, as well as expanding to further types of variation and non-coding elements, and incorporating proteomics and other omics data. So we're very excited about this data set. And we hope that this gives you a taste of the data that will be hopefully coming your way soon. And I'd like to end by thanking the head of the Centre for Genomics Research, Slavoj Petrovsky, and my colleagues who have led and played a key role in the work that I've told you about. And finally, no UK Biobank talk would be complete without thanking the wonderful participants who have gifted us this amazing resource. Thank you.
So Katherine, thank you very much for a terrific talk and for stepping in at short notice in place of Katherine. But really good to see some very real examples emerging about the added value of the whole genome sequencing data, the things that you can get that you can't get from the whole exome, and also some of the rationale for doing the reprocessing using DRAGON to give the increased precision, but also some of the additional outputs. I'm sure we can talk about that more in the panel discussion. So we'll hand over now to our final speaker of the session, Roland Ealing, who's the CMO for AWS, who together with DNA Nexus have been really key to supporting the whole platform endeavor. And Roland will come and share his thoughts and AWS's thoughts on equitable health research. Thanks, Roland.